Hello everyone, welcome back to my channel. I'm Tepin and today I'm going to show you how to submit your mitochondrial genes or any protein coding genes. So first and foremost, you must have your gene. This is my gene. I have actually downloaded it from NCBI just to show you. I'm not going to submit it actually. So this is Drosophila melanogaster cytochrome oxidase subunit 1 partial coding gene, partial in both 5' and 3'. And I don't know the reading frame, but I will try to do it manually. So first, go to NCBI website, and then you will see something like this. Then press submit, then go to this submit, and then click new submission. Then press eukaryotic because your organism is eukaryotic, and then click this one, any other features. And then you have to choose original because it is your own data. So I'm just using original for now as an example. You can choose submission title. For example, I'm just going to click use this. For example, you can put your specimen number, then enter your details, and then choose the method by which you got the sequences. For example, Sanger sequencing, and you should have assembled sequences. It's better. Continue. Then you have to choose a specific date, and then you have to know which DNA that you are submitting. Any three of these. So you can choose mRNA for now. And we know we have mitochondrial genome. Are these complete mitochondrial genome? No, because it was partial. So yes, it's linear. We can now choose our file here. Continue. Press continue. Again. Now they will ask you what is your source. Isolate, strain, clone, cultivar, whatever it is. You can just choose it. For now, let's just choose isolate for now. Continue. And then organism is Drosophila melanogaster. And then choose your date of collection, geolocation, and isolate. For example, this is my isolate. This is just for example. And then we can continue. And then now this is the crucial part here. If you have the features, you can upload it, protein FASTA or whatever you have at CDS feature coding region. This will be much faster if you know the coding features. But if you don't know, then add features by web forms. You can click this, and you know your protein DNA sequence code some kind of protein, right? So you can click coding protein sequence, then add CDS feature. You can choose entire length. If you know specific sequence, then that's good. If you don't know, then you can choose entire sequence. It will choose the entire sequence that you're submitting, and then you should know which plus or minus strand. For this, I've already explained it in my previous video. For now, I know this is plus strand, so plus, and then features in incomplete at both 5' and 3'. I know that, and I don't know the reading frame, so I'll first choose one, and then protein name is cytochrome oxidase subunit 1, and gene symbol is COI. You can add more if you have like splicing and other features, but for now let's just accept like this. So you can see it says internal stop codon were detected in the protein translation, which is biologically incorrect. Provide a correct sequence on the sequence page, or provide more information to the correct the CDS annotation. So if you are sure that your sequence is correct, then you can just change the reading frame. Let's just choose 2. And you can also see here, this is the translation. This is the protein that is coding here. But in this case, you can see that The protein that is coding here are these protein. So I'm going to choose reading frame 2 and then click accept again to see if this coding, if this code is the same protein. You can see they have again detected stop codons, and it is coding some other protein. So I will choose the third reading frame and accept. You can see it is detected correctly. The protein codings are also similar. You can see here. You can then click continue, and then you can put all this information that you are, since you are publishing, so you should have some details, right? You then use all these details. Reference title you can add, or you don't even have to add. It's not a big problem. You can just click here, and then they will generate this. You can see Drosophile has isolate this and this and this, and the features are here and all those here. You can see. And after this, you can just press submit here, and then your submission will be done. And then the NCBI will review, and if your submission is correct, they will provide a session number. So this is how you submit your mitochondrial gene, protein coding genes. And if you have any further query, please comment. And thanks for watching. Please like, share, and subscribe. Thank you.